Hi, everyone. Welcome to today's Altasciences webinar on overcoming bioanalytical challenges for PK/PD assessments in phase 1 biologic studies. My name is Dr. Dave Nguyen, and I am the General Manager and Medical Director here at Altasciences. Again, I'm Dr. David Nguyen. I'm the General Manager and Medical Director here at Altasciences. I've been here since 2017, and I'm an anesthesiologist by training.
Hello, my name is Len Lesotar. I'm the Vice President of Laboratory Sciences at Altasciences. I've been with Altasciences for 2 years, but have over 15 years of experience in the contract research organization, specifically in biologics development, as well as 6 years experience in the National Research Council of Canada developing biologics and biotherapeutics.
Today's learning objectives are as follows. We're going to take a look at the integrated advantage for a successful biologics and bioanalysis for first-in-human trials. We're going to consider the effective preclinical to first-in-human bioanalytical assay transitions that would make it effective. And finally, we're going to take a close look at a case study demonstrating the impact of using the integrated advantage model. Through some of the considerations for a successful early phase trial looking at these biologics. And before we get started, let's all get on the same page with some very clear definitions of what we're going to be discussing today. The bioanalysis portion involves the identification of substances as drugs, metabolites, proteins that are part of a biological matrix to obtain a quantitative measure for the study of the PK, the toxicokinetics, and even bioequivalence. And what are biologics? They're substances whose active components are derived from a biological system. They include any of the following things we have listed here, from monoclonal antibodies to fragments of the antibodies to bispecific antibodies to antibodies that actually bind to drugs and conjugate and have their effects. They can be very sophisticated engineered proteins. Right now, we're seeing a lot of development in T-cell directed therapies and CAR T therapies in the cancer arena. Gene therapies and genetic medicine gets a lot of attention, especially from this arena. And of course, right now, living through the pandemic, vaccines are all considered biologics. What are first-in-human trials? Well, they're pretty much exactly what they are called. FIH studies are a key step to developing medicines that have been thoroughly tested in preclinical animal models and now being administered to people for the very first time. Generally, when we conduct these first-in-human studies, we're going to be using healthy volunteers who are not patients. On the right of this slide, there's a comparison between small molecule chemical drugs, generics, if you will, versus the biologics and biosimilars that are looking to develop new biologics based on previous models. You can see that they're very, very different. And the 3D rendering of that biologic molecule is a testament to how complex and just generally much bigger they are compared to the small molecule chemical drugs that we see. We're going to take another closer look at these differences later today. But for now, that's a snapshot of what the differences are. So here's a closer look at the differences between the biologics and the small molecules. Like we mentioned before, biologics are large, they're big on a molecular level. And the term bioequivalence generally implies discussing it amongst the small molecules. When it comes to biologics that are so complicated and sophisticated, complex, and large, it's really impossible to guarantee bioequivalence with the larger molecules. And so the term biosimilar is generally used when discussing biologics. The specific division of the FDA that governs the regulations of the development of biologics is the CBER division, whereas the small molecules are governed by the CDER division, and they apply their regulations to small molecule development and peptides. And again, bioequivalence means every single batch that is synthesized must be identical. And this can be accomplished with small molecules because they're simpler. They have a very well-defined solubility, molecular weight, melting point, et cetera, et cetera. And so every batch needs to be equivalent to each other. And it's easier to show that each batch is identical. The distribution of small molecules can span the entire body, all the water content. And for these DE types of studies, there really isn't a need to demonstrate as a high N. 12 participants might be enough to show equivalence between two different small molecules. That number generally has to go higher when we explore biosimilar studies with biologics. Here's a chart that really just expands on the biggest differences between small molecules and biologics. And you'll recall in a couple of slides earlier, we saw a 3D model rendering of the real size. And so if we look at the actual number, small molecules really are molecular and less than 1 kilodalton, whereas the biologics are generally much, much bigger and always bigger than at least 1 kilodalton. Small molecules work in a very simple manner. There's a ligand, and binding to the receptor to enact an action, whereas biologics are much more sophisticated in the way that they cause their ultimate reactions, whether it's changing in the protein structure and folding, denaturing proteins, substituting different amino acids in the protein itself, so on and so forth. You can read it all there, much, much more sophisticated of an action. Small molecules can be administered in many different ways, whether it's topical or parenteral through an IV infusion or just through the digestive system orally. Whereas with the sophistication of a biologic and their size, it requires generally an IV administration. The pharmacokinetics between these two molecules are also extremely different. The Tmax for a small molecule happens in just hours, but with a biologic that has much longer Tmax in days. The distribution too within the body we just discussed, the small molecules can diffuse pretty thoroughly throughout the body, whereas biologics by nature of their size have a very limited distribution. And this, of course, has a lot of effects downstream in terms of analyzing the PK, the PD data, and the bioanalysis of it. The diffusion, the reason it's distributing differently with small molecules, it can be active or passive. It's not hard to get these small molecules dispersed throughout the body. Whereas with biologics, you're going to require some transcellular, paracellular active transport here to get them to where their place of action will ultimately be. Subsequently, the elimination of these products out of the body is very different, like night and day. When we're looking at small molecules, the liver and the renal biliary system, the traditional systems of excretion of drugs out of the body are certainly in effect, but with biologics, there's an added layer of complexity requiring intracellular lysosomal protein degradation to break down these products so that they can ultimately be eliminated in the body via the other traditional routes as well. And so because of that extra layer of complexity, we see that small molecules behave in their elimination process in either a linear or nonlinear way. Linear, the traditional way, assuming that we have a fully functioning and effective hepatic and renal excretion system. But when looking at biologics, it is not a traditional linear elimination because of the extra requirements to degrade the products and eliminate them from the body. That's also manifested in the half-life of these different molecules, with small molecules being the traditional less than 24-hour half-life. We don't see long-acting small molecules in play, whereas with biologics, they can last and have a half-life that is much longer, days to weeks. And finally, something that is going to be very unique to biologics is immunogenicity. The human body generally will not develop antibodies against small molecules. They're just so small that they don't attract the attention of the immune system. Whereas with biologics, they're large enough to cause the production of anti-drug antibodies. And those antibodies will be a topic that we'll discuss in a bit here. But what is the use of biologics in humans? Why do we have them? What's their purpose? So the overall goal of biologics is really personalized medicine. We talked a little bit earlier in one of the opening slides about genetic medicine, and obviously, gene therapies that are, specifically targeted for specific phenotypes and genotypes. And so the use of these therapeutic proteins, i.e. biologics, whether they be monoclonal antibodies or any of the other ones, are to neutralize elevated antigen levels or any overexpressed disease targets so that we can return those levels to before any disease could be manifested or eventually produced. That's the purpose. So what are the early phase considerations when designing a study looking at biologics, especially in a first-in-human trial where we're studying biologics in people for the first time? And the first thing to consider, of course, is the dose regimen and optimizing that dose. Generally, a lot of data has already been generated from the preclinical animal studies exploring what the maximum dose of biologics can be. And then when transferring that over into humans, as a rule of thumb, the dose generally has to be divided by a factor of 100 just to start. And when starting in humans, sentinel dosing is critical. A very, very small group of participants to explore any adverse events that might be generated from these biologics. Given that even though a lot of preclinical data has been created, it's still an unknown how the human body will interact and any effects, unwanted effects, off-target signals, safety signals effects that could be generated from these biologics. And so the sentinel group is really critical as part of the dose regimen optimization. The clinical pharmacology issues in biologics development. What are the drug-drug interactions that these biologics may have with each other? So what are monoclonal antibodies going to interact with other monoclonal antibodies or any of these other biologics? Or biologics interacting with small molecules? What does that look like? How does that affect the PK, the PD data, the bioanalysis that will be generated from this type of study? A safety concern with QTC prolongation is not really the biggest concern, and so waivers can be granted to avoid looking at this specific safety marker. But immunogenicity is really one of the key factors in considering biologics and safety data. And it's not just safety. It is about whether or not these anti-drug antibodies have any effect on the efficacy of the drug, as well as an overexpression of an immune effect after the drug is given. So we'll talk about that in a little bit more in detail in the coming slides, but certainly a highlight for biologics drug development. And given the complicated, sophisticated biologics that are produced and the different effects that could occur in the human population, of course, there's different populations to consider, whether it's certain ethnicities with certain expressions of genes that aren't found in other populations, a simple gender differences between men and women who may or may not express certain proteins that may be impacted by these biologics. And the age group of the population. How does this manifest, if any, effects and any safety signals in the young versus the old? Is the efficacy different based on whatever genes are being expressed throughout the course of our life? What does that look like? And finally, PBMC cell isolation, harvesting, a very, very intricate and complex bioanalysis that needs to take place to really get the big picture of what the biologics are causing in the human body. So we talked about the starting dose and the dose regimen optimization and why it's so critical for biologics development. When we're looking at small molecules, this is the model that's generally followed. No observed adverse effect level. It's very sensitive to the species or however it's clinically relevant, and then converting that to the human equivalent dose, going from preclinical to clinical. You take the NOAEL and you divide it by 10 to find the starting dose in humans. And that really is the maximum recommended starting dose, is a fraction of the human equivalent dose. So contrast this with large molecule biologics. So in biologics, just new terminology, different terminology, just like bioequivalent, biosimilar. Here, it's the minimum anticipated biological effect level, MABEL. And it's usually much lower compared to what we looked at for the small molecules. And like I said earlier, you divide it by 100, a factor of 100 versus 10 for the small molecules to start. And these are the reasons why, biologic agonists can cause downstream effects that we're not aware of, that might be not seen in animal models. There could be cytokine storms that develop from these biological agonists that we would only observe from a human study. And so we would want to be aware of any of these signals before a higher dose is given. And also mentioned earlier for the sentinel dosing to really explore that before we go to a larger cohort. And it can depend on both in vitro and in vivo studies, humans, animals, but usually the idea is to be very conservative. There's a saying that if you have more than a 20% have a reaction, the study needs to be stopped. But if it's less than 2%, we're okay to continue. So very, very conservative. But the exception here is a unique one. You may recall in the beginning of my part of the presentation that I mentioned that these first-in-human studies generally start with normal, healthy participants who do not need the biologics, the therapies. However, here, there is a potential for starting these studies in the patient population who might actually benefit from these biologics. And the idea here is that the benefits outweigh the risks. The risks of having these downstream effects, these side effects, these cytokine storms. Well, if someone is truly in need of the benefit of these therapies, then it could justify starting the study in a patient population, accepting a certain percentage of potential side effects if the patients can benefit from this therapy. Finally, one of the things that I've mentioned previously and multiple times that we're going to talk about here is the immunogenicity that can develop against biologics. They're big molecules, and so our bodies can potentially develop antibodies against these biologics. These anti-drug antibodies, ADAs, need to be considered. So previous exposure to biologics. A participant who has participated in a biologic study in the past could have developed anti-drug antibodies to biologics. And now if that same participant returns and participates in a different biologic study, well, now you've got two very complicated exposures to sophisticated biologic proteins, and how their ADA will react to the new biologic is a concern. It can also make it difficult for the bioanalysis portion to understand, is the development of ADA to this current biologic, or is this an exposure that has already occurred and now we're looking at an ADA that was formed previously, and the currently studied biologic isn't the one that's causing the development of these antibodies? So the challenge on a bioanalytical side and on a clinical side, these ADAs can manifest as rashes, immune reactions, cytokine storms, your adverse events that we're looking for here in the clinic. Is it this study and this molecule, this large molecule, that's causing these adverse events? Or is this a participant who is coming back to us and now having been exposed to two, three, or who knows, maybe even more biologics in their history of participating in studies? Is this reaction a result of the congregation and the accumulation of all these biologics over the course of their time working on studies? And so it really clouds the picture. These ADAs can also make it difficult for the current biologic that was just administered to produce the effect that is being designed for and the therapy that it's looking to treat and use for. So this is not something that we want in our study participants. And so here at Altasciences, we do make an effort to keep a patient database or a participant database that is very clear and organized on who has participated in biologics, large molecule studies, and who is biologics naive, if you will, and has not been exposed before, so that we can really avoid all these complications and things that would cloud both our bioanalysis and study data, as well as on the clinic side here with me, some of the safety concerns that could arise from these therapies. I've mentioned multiple times that sentinel dosing is critical. Lower starting doses are essential. And just like when we touched on in the past, the follow-up for these studies is much longer compared to small molecules. We can go up to a year with especially these vaccine studies, which we are all very familiar with as we're living through right now with the booster shots that is being approved and authorized and so forth. So, and like I mentioned before in my previous slide, cell isolation and PBMC is really critical and necessary. And here at Altascience, we do have the capability of doing that here on site for the modulation and tracking of the immune response to biologics in the study participants. It's critical. Bioanalysis, it really is for first in human studies. This is going to be my last slide as I bridge over to Lynn's. Just a few definitions to set the stage for her portion of the presentation. But as some of our audience members will be very familiar with, these definitions transcend both small molecule bioequivalent as well as large molecule biosimilar studies. You've got single ascending dose, where the primary endpoint is to look at the safety of the compound after a single dose. Some other secondary endpoints could include looking at the PK data to look at Cmax, Tmax, half-life, et cetera, and exploratory endpoints could be looking at the pharmacodynamics as well, and its effect on the body and the participants' other lab values. And then, of course, the other side is the MAD, the multiple ascending dose, looking at the safety of the compound after multiple dosing. So looking at the PK as a secondary endpoint again, looking at the peak and trough values after a steady state is reached, looking to see if accumulation occurs or if there is enough of an elimination to really maintain that steady state. And it's very common as an additional endpoint to look for the pharmacodynamics as well, especially after maintaining that steady state. So proof of concept, does it work? Yes or no? And with that, I want to thank you for listening to my portion of the webinar, and I'd love to turn it over to my colleague Lynn to walk you through the bioanalytical side of biologic studies.
Thank you, Dr. Dave, for your presentation, and to all for attending today's webinar. Today, I will be presenting the integrated advantage for first-in-human biologics bioanalysis. My name is Lynn Lesauteur. I'm the Vice President of Laboratory Sciences at Altasciences. So I'd like to start off by describing what is the integrated advantage for first-in-human bioanalysis. It is the leveraging of bioanalysis expertise in a one-stop shop to effectively transition from preclinical to first-in-human studies. The specific transition point we're talking about today is highlighted by the green oval. Now, effective transition depends on many capabilities, including the bioanalytical testing from non-GLP through GLP, non-clinical safety testing facilities and expertise, clinical pharmacology and clinical efficacy facility and expertise, as well as supporting research services, which include project management, pharmacokinetics, biostats, data management, regulatory, and medical writing. Successful bioanalytical transition depends on operational and scientific knowledge, as well as regulatory guidance and expertise. All of this is enabled by a single point of contact for all services, which is the program manager. And all these teams and folks use specialized tools such as customized reports, centralized scheduling that captures the whole project components of the programs, detailed timelines for deliveries, as well as personalized dashboards, which capture client-specific requirements and preferences. What are the impacts of the integrated advantage? First of all, it effectively transitions bioanalytical methods from preclinical to clinical studies, which ultimately leverages know-how, saves cost, and time savings. Secondly, we move seamlessly from the clinic to the lab and back and forth, and of course, data services for rapid analysis, interpretation, and issue escalation if need be. More accurate planning and the right assays at the right time is also the result, including more proactive experience with the necessary critical reagents and their management. We also improve agility to adapt methods and adjust timelines based on the needs and experience within the program. And we strengthen client relationships with the continuity of contacts that results through this integrated advantage. Ultimately, the risk analysis depends on quality and timely pharmacokinetic and pharmacodynamic data that is enabled by the integrated solution. Regulatory knowledge allows effective bioanalysis. In this slide, I would like to highlight key changes in regulatory bioanalysis of biologics. So if we start off by thinking 20 years ago, the types of biologics in development were mostly small molecules, and the regulatory status for development of these biologics was really at its infancy. There was no bioanalytical method validation guidance, no standardization of this, and we were at the infancy of immunogenicity and immunotoxicity testing needs for biologics. Whereas now there's many types of biologics in development, including monoclonal antibodies, antibody fragments, and iterations of those, engineered proteins, and gene therapies. And of course, now the regulatory status has evolved. We have bioanalytical method validation guidance, immunogenicity and immunotox testing guidance, as well as specific white papers that are product specific and also address causes for concern with biomarkers and critical reagent qualification and validations. In the future, there will certainly be even more targeted therapies with novel approaches, new methods, and instrumentation, as well as patient-centric sampling. Thus, the regulatory framework is evolving. The current bioanalysis regulatory know-how is highlighted on this slide. So the details of the bioanalytical method validation guidance are here. There's published guidance from the EMA, the FDA, and ICH harmonization. There's specific guidance on immunogenicity testing from both the FDA and EMEA, as well as immunotoxicity testing, a guidance, an example of which is shown through the S8 immunotoxicity studies for human pharmaceuticals. There's certainly also several recommendations with regard to biomarker qualifications and validations. A key document to refer to is the CEDAR Biomarker Quantification Program and Framework. And this all also relies on the CRO expertise in good laboratory practice and application of those guidance, as well as good clinical practice, standard operating procedures, validation, bioanalysis and study plans, and the regulatory inspection history that the CRO has. The evolving regulatory landscape for biologics is highlighted by recent white papers, which highlight recent issues in bioanalysis. For example, gene therapy points to consider, biotherapeutics and stability challenges, qPCR assay validation, flow cytometry, and ELISpot validation considerations, as well as application of hybrid assays that include ligand binding assay and mass spectrometry, and new techniques such as microsampling. Challenges for biologics development include applying bioanalytical regulations and know-how. Since bioanalytical method complexity and regulatory expectations increase throughout development, this is certainly a challenge. For example, in early phase discovery of biologics, the bioanalysis consists of early stage assays for appropriate pharmacokinetic and pharmacodynamic analysis, and these assays are mostly fit for purpose. When one transitions to the preclinical toxicology GLP studies, they need to have validated assays for toxicokinetics, anti-drug antibodies, and immunotoxicology testing, as well as fit-for-purpose assays for biomarkers. Now, the critical transition point between toxicology studies and the first-in-human studies is what we're going to be focused on today, and has additional complexities. So if we start off with the validated PK assay, this is certainly a requirement and has to be carefully considered with regard to, can one transition the method that already exists preclinically and simply adapt it for clinical use, or do you have to adapt a brand new method? There's certain considerations such as sensitivity, specificity, and measuring total versus free drug that is also relevant to consider. Anti-drug antibody assays are also required to be validated, and these also present distinct challenges from the preclinical toxicology assays, because now we're dealing with patient and matrix specific considerations that are novel, as well as need for further characterization of epitope specificity of the immunogenic response, as well as full characterization. Neutralizing antibody assays are also required at this stage in development and need to be validated. Certainly, the panel of biomarkers tend to increase at the stage of first-in-human testing and can include biomarkers for target engagement and exploratory purposes, which are developed on a fit-for-purpose basis. And if there are inclusion of biomarkers that are used for confirmatory aspects, then full validation is required. The case study I will be presenting today is the effective bioanalytical transition from preclinical to first-in-human phase 1 single ascending dose study for the safety, pharmacokinetics, relative bioavailability of an immunomodulatory protein. Specifically, it is a monoclonal antibody that binds a soluble protein. I will be speaking about general considerations for effective transition, plasma pharmacokinetic assays, serum anti-drug antibody assays, blood for pharmacodynamic assays, including introduction of novel techniques such as microsampling. One of the first considerations is the decision of what bioanalytical platform to use. Key considerations are which component of the drug should be quantified, how can pharmacokinetics and pharmacodynamics be best characterized, and which platform has the required sensitivity. In this case study, ligand binding methods were used for these assays. However, I would like to emphasize there's more and more use of mass spectrometry for biologics quantitation because of its exquisite selectivity, and often these two assays are used in conjunction to answer appropriate questions. So let's start with some general considerations. The first is to focus on logistics. And these require key planning documents, such as comprehensive program and project management plans, communication plan, dose escalation and safety management plan, as well as risk assessment and mitigation plans. Secondly, focus on bioanalytical data deliverables and expectations, including clarity on what is due when from all the stakeholders, including details such as sample shipment, analysis, and data release, and which data will be used for safety meetings and when is that required, as well as the action plan for sample repeats and additional sampling. PK studies and all other endpoints can change very quickly. These changes can be fast and additional sampling can be required. The agility between the first in human site and the bioanalytical teams is key to enable appropriate decision making. Bioanalysis for pharmacokinetics presented challenges that we were able to proactively solve using the integrated advantage. In this study, the preclinical format for bioanalysis for the PK was ELISA, whereas in first in human, it was a mesoscale discovery based platforms. Key challenges in this change involved a need for increased sensitivity as well as observed matrix interferences between the preclinical and first in human studies. The benefit of the transition within one team was the savings of time due to experience with different platforms and critical reagents. Specifically, with regard to the matrix interference, it was solved by maximizing the antibody pairing that was used in the human studies compared to the preclinical studies, resolving the selectivity issues, and careful analysis of the different matrix components which interfered with capture and detection methods. A thorough evaluation of parallelism during method development to determine sensitivity to the endogenous analyte, selectivity, and minimal required dilution was performed. Well-established procedures for critical reagent qualification that were in place from preclinical to clinical certainly increased the speed and development of the appropriate clinical bioweigh method. Increased sensitivity was achieved by changing platforms from an ELISA to a mesoscale discovery platform. Bioanalysis for immunogenicity assessment was a challenge in this study as well. The bioanalytical assay types included were anti-drug antibody measurements and neutralizing antibody measurements. Preclinically, the ADA response was measured by ELISA, as well as in the first in human studies. The challenges included critical reagents, matrix changes, target interference, and increased sensitivity requirements. The experience with the one-stop shop with preclinical assay and management of clinical reagents saved time. With regard to the neutralizing antibody assay, it was not a requirement preclinically, but it was incorporated within the first in human study and it composed of a cell-based assay and full characterization. The benefit was having an integrated report throughout the program of the immunogenicity of the compound and its repercussions. I would like to further comment on the critical reagent issue that was solved. Changes of assay critical reagents when selecting format was species dependent. So whenever possible, use a non-species dependent assay format. Early discussions with the client in the preclinical section of the program minimized the assay format changes between preclinical to clinical studies. Secondly, matrix challenges were solved due to proactive knowledge of the challenges that would be encountered in the clinic, such as adding complexity of disease type populations for cut point, specificity, selectivity, and sensitivity, contributed to additional interference and result in complex cut point analysis. Our advantage certainly was the ability to obtain pre-treatment samples from appropriate subjects. Target interference was also solved. Sometimes the drug target is not present in the animal model preclinically, but will be in the human subject, which can result in interference. Thus, we ensured target interference is evaluated using exogenous target in the non-clinical phase of the method development. This will better support the transfer to the clinical phases. Increased sensitivity was also a requirement. Higher sensitivity is required in the clinic versus non-clinical studies, and we were able to save time by ensuring the assays developed in the non-clinical phase can still meet the clinical sensitivity requirements. There are additional challenges with regard to bioanalysis for immunogenicity. Specifically, 80% of the non-clinical assays consist of only screening and titer assays, whereas confirmatory assays were required once in the clinic. In confirmatory assays, samples are tested in the screening assay that are positive are subject to confirmatory assay to demonstrate that the anti-drug antibody response is specific to the product. Secondly, NAB assays are required in the clinical studies, whereas in preclinical studies, they're generally not included. The NAB assays consist of in vitro neutralization assays to indicate the potential of the anti-drug antibody response to inhibit the therapeutic activity of the product. And thirdly, binding specificity, which is not a requirement in non-clinical studies, is required in the clinic. For example, a drug with an endogenous counterpart or multi-domain protein may require that specificity of immunogenic response be evaluated. And lastly, ensure appropriate positive control generation is started early and is appropriate for product development. Additional challenges include additional considerations for matrices, such as whether a co-administered drug with similar homologies need to be evaluated for possible interference. There may also be interference with rheumatoid factor that needs to be evaluated in certain patient populations. And there may be presence of pre-existing antibodies that would have to be taken into account. In addition, critical reagents cannot be underestimated. They need to be secured for the duration of the long-term studies for management and qualification and retest dates throughout the course of the whole program. And to make sure to adapt the labeling strategy specifically for the conjugated molecule to support long-term studies. For example, if they're stored at minus 80 with an ongoing stability from the non-clinical stage of the program. Certainly, accumulated data and experience with critical reagents will help to better plan the clinical phase. Bioanalysis for biomarkers also had challenges in this study, which was mitigated by the integrated solution. There were many types of biomarkers that were included. In preclinical, for example, exploratory cytokines were included, as well as in the first in human study. And there were several kit issues that had some challenges. The benefit of the integrated solution was the scientific and technical expertise with the assay and its purpose saved time and cost. Immunophenotyping was also included preclinically and in first in human. Challenges included PBMC isolation at the clinical site, as well as antibody changes and requirements. The benefit was existing experience with platforms, reagents, and data handling led to greater efficiency. Specifically with regard to the kit issues that were solved, kits and antibodies can sometimes not be appropriate for both clinical versus preclinical analysis, and assay ranges and sensitivity improvements are needed. As well, one has to consider endogenous levels of biomarkers that can vary between species. And thirdly, the reagent supply issues during COVID specifically and mitigation and providing alternatives was critical to the not delay any time deliverables within the study, and planning for lot to lot variability and comparability assessments, including the ability to procure critical reagent antibodies and appropriate vendor qualification. The PBMC isolation was established quickly at the first in human site, enabling the appropriate transition and sampling within that study. The assessment of appropriateness of antibodies within the immunophenotyping was also conducted. And I'd like to just say a few more words about the biomarkers. Although I've emphasized the exploratory biomarkers, certainly there was a whole panel of additional biomarkers that were included, and there were multiple discussions with the client to enable and understand the appropriate regulations that would needed to be applied for whether they were exploratory or confirmatory biomarkers. Ultimately, when biomarker data is used to support a regulatory decision-making process, such as pivotal determination of safety and or effectiveness, or to support dosing instructions and product labeling, the assay should be fully validated. The last example in this case study is leveraging the transition between preclinical and first in human by including new techniques for bioanalysis, such as microsampling. Microsampling is the collection and analysis of small volume matrix samples. Microsampling enables frequent sampling that will have better therapeutic dose monitoring and could be particularly important for biologics monitoring. These novel analytical methods have been recently developed, and the advantages include allows collection of clinical samples from remote locations, for example, non-ambulatory disease populations and pediatric population. It also frees up blood volume to collect additional and or alternative samples, be it more time points or additional biomarkers. It also provides patient convenience and enables cost savings through ambient temperature sample shipment. There are several bridging considerations between preclinical and first in human with regard to introduction of new technologies. And the first place to assess is the bioanalytical method validation guidance. The FDA certainly encourages the development and use of new bioanalytical technologies. However, use of these two different bioanalytical techniques for the development of a drug may generate data for the same product that's difficult to interpret. This can occur when a second platform generates drug concentrations differing from those of the first. Thus, data produced from the second platform must be bridged to that of the first one. The sponsor wanted to include microsampling within this study, specifically for the PK endpoint, which was an LBA-based endpoint. So preclinically, we use sample venous blood sampling, whereas in the first in human, we wanted to apply the microsampling using dry blood spot technology. The challenge was the change in technology for sample processing. The benefit of using the integrated solution was familiarity with both methods and a one-stop shop for preclinical and clinical bridging, which saved time and cost. This was accomplished by proactive bridging with incurred sample reanalysis and additional validation of microsampling approach prior to use. Careful analysis of the bioanalytical method validation guidance and end use of the data to establish specific plans and logistics and workflows and SOPs was critical, and thorough validation plan with assessments and acceptance criteria, including storage and handling temperatures, homogeneity of sample spotting, hemocrit, stability, carryover, and reproducibility. This then will enable further clinical studies. I hope you have learned the following take-home messages from this presentation: that leveraging bioanalysis and a one-stop shop to effectively transition from preclinical to first-in-human depends on the teams, the tools, and the expertise, which will then lead to the following: time and cost savings, better relationships, and effective implementation of innovation. So I'd like to thank the audience for the great questions received. I had to select just a few, so I'd like to start with the first question for Dr. Dave, which is, what are the most common challenges in first-in-human studies with biologics bioanalysis from a clinician's perspective?
Oh, wow. Thank you, Lynn. There are certain challenges for sure from the clinicians and medical safety side of conducting these first in human trials, and biologics specifically. I've gone over a lot of these safety concerns in my portion of the slide, but really to highlight, in addition to those pesky anti-drug antibodies that have both complication to the bioanalysis, but also the medical safety issues that can arise and cause adverse events. That's probably the main one, I would say. But here at Altasciences, as I sort of mentioned earlier, we do have a participant database that marks which participants have worked on studies and have been exposed to biologics. And so we can filter those study participants out of the equation when we are recruiting for specific trials and really target biologics naive participants and have really extensive marketing outreach programs, even to clinical trial naive participants too. So that can help to mitigate that. And then really the PBMC cell isolation harvesting that occurs in these trials requires a lot of technical expertise on the collection side. So a team of phlebotomists that are trained, that are familiar with the number of tubes, the volume of blood that needs to be collected, and then having an on-site cell isolation lab to prepare the samples properly and extract them for shipment to go to a bioanalytical lab is really critical. And we enjoy that here at Altasciences because of the work we do and how often we do it, that the teams are familiar enough with this process to make it successful.
The second question from the audience, Dr. Dave, is from a practical perspective, what are the challenges you most often see with regard to obtaining timely bioanalytical data?
Yeah, thanks, Lynn. And it's challenging because for us here as a site, oftentimes we will ship off the samples to the bioanalytical lab somewhere else in the country or even internationally. And just the logistics of time that it takes for the samples to make it there, and then the communication of whether or not those samples are viable or adequate or any variation thereof. It just takes more time, and it's hard for us as a clinical site to adjust our practices, or in certain cases if things need to be repeated. uh, let's say, for example, a cohort comes and they dose and we collect the samples and we ship it off. The time that it takes for us to get constructive feedback as to whether or not those samples are good is just too long. The cohorts have moved on, the participants have entered other studies sometimes. And so having a solution that is in-house, that actually a lab that uses our same labeling system, our same inventory system, a communication within the same organization, so that we get those feedback rather quickly, allows the site, the clinical site, to make a course correction, repeat a cohort if necessary, or change a practice so that the success of the trial isn't jeopardized. Okay, I've got some questions here for you from the audience. Can you comment on any critical reagent managements from the preclinical to first in human studies?
Thanks for the question, Dr. Dave. I'd like to emphasize that critical reagents are critical, obviously, for the bioanalytical method development. They're the cornerstone of the appropriate method. Poor quality of these critical reagents leads to unreliable results, delays, and loss of time and money. So the more de-risking and planning and mapping out of the critical reagent plan, including how and when new critical reagents will be generated, what would these future lots be, and the characterization required, as well as the qualification of these critical reagents within the bioanalytical assay. And the supply chain and storage and distribution of these is absolutely necessary to map out internally with the sponsor and with the CRO and with all the stakeholders in the program, allows to effective transition. And a partner such as Altasciences, who can manage the critical reagents throughout the development, is critical.
Thank you, Lynn. It makes perfect sense. And for the second question here, we have, what do you see are the major challenges facing novel biologic modalities that transitioning from a preclinical to a clinical form from a bioanalytical perspective?
Yeah, that's a good question. There's so many different types of biologics now in development. I'd say that some of the major buckets of different challenges. Number one is the lack of guidance documents, thus alignment of the plan based on scientific and regulatory understanding with all the stakeholders is really the most important starting place. And I would add to not underestimate the time needed to develop and validate the appropriate bioanalytical method to ensure that you're asking the right question with the right method for that right novel biologic. And an experienced partner will expedite the drug development program.
Thank you, Lynn. Planning going a long way here, right?
Absolutely. Thank you, Dr. Dave. And I'd like to mention, please do not hesitate to contact us should you have additional questions or would like to discuss your specific program needs.
Thank you, Lynn, for answering all those great questions. And that concludes our webinar today. I want to thank you for your attention to my part of the presentation. And if there's any questions from the audience that wasn't covered today about study design, safety monitoring, and biologics from a clinical standpoint, please do not hesitate to reach out to me. Thank you very much.